Okay, everybody. So today we're going to talk about DNA replication. Mr. Mandaraki talked to you yesterday about DNA structure. So the important thing is, how do we make DNA? Why do we need it, and when do we make it? So DNA replication is the process of copying DNA. We need to copy DNA before mitosis and before meiosis, which we haven't learned about mitosis and meiosis yet, but those are the processes of making more cells. So we go through DNA replication before we make new cells. Without DNA replication, our new cells wouldn't have any DNA, which we know all of our cells need the instructions for life. So the first step of DNA replication is that the nucleotide chains unwind and are separated by a helicase. And helicase is an enzyme. The helicase breaks the helix or splits the helix. So remember that the DNA strand is in a twisted ladder form, which you see in the picture there. And the helicase comes in and takes the twisted ladder. It unwinds it like this and then unzips it so it opens up. So literally it starts to look like a zipper. So the nucleotide chains have to unwind. Those hydrogen bonds between the nitrogen bases have to break. And then they separate to make room for the new nucleotides that are going to be added into the molecule. So after the helicase comes in and splits the helix, the DNA polymerase, which is also an enzyme, remember back to the enzyme unit, we said that lots of enzymes end with the letters A, S, E. So DNA polymerase is an enzyme that bonds to those open strands. So now your helix is open like this. The DNA polymerase comes in and bonds to the open strand, one end here and one end here, and they sort of work opposite each other and meet in the middle. The DNA polymerases are then going to add nucleotides to each of those strands. So I'm going to play this short video here so you can see, and I don't know if you'll be able to hear the sound, but I'll narrate through it for you. Maybe. There we go. Sorry it's repeating. Oh, goodness, this is not going well for me. Now you see the DNA polymerase is going to come in here and add the nucleotides. So I'm going to, okay, well, finally let me pause. So if you look here, not at that, if you look here, you start to see the open strands, and it's going to slowly add those nucleotides in. This is your DNA polymerase here. It's going to add those nucleotides in, creating a new strand of DNA on both sides. This is called semi-conservative replication because you get half of your strand is old, the original DNA, and the other half is new DNA, so new nucleotides that are added into the DNA molecule. So here's just a still image of that. Here's your DNA molecule. The helicase comes in and unwinds the DNA strands and creates what we call the replication fork. So the DNA helicase comes in, it unwinds those strands, separating them. The DNA polymerase then comes in and adds nucleotides. And this picture is a little bit misleading because it really happens from two opposite directions. And like I said, one starts at the top, one starts at the bottom, and they sort of meet in the middle. And then when you're finished, you have two brand new strands. Half of the strands are old and half of the strands are new. So DNA replication is extremely accurate. There's only one error every 10,000 base pairs, which means that mutations or changes that occurred actually don't occur very often. And so during that replication process, it's continually proofreading and repairing whenever possible. So it's going to add a nucleotide and check that it's correct, add a nucleotide and check that it's correct. If there are errors or mutations that occur during that process, it creates a mutation which can change the nucleotide sequence, which can then change the proteins that are going to exist within that cell. That can end up causing problems like disease or mutation or, developing or creating too much of a molecule or not enough of a molecule. So DNA replication, while it does happen really quickly and it happens in every single cell, so it's happening on a very large scale, it's extremely accurate. Thank you so much for watching my short notes on DNA replication. And if you have questions, make sure you ask them at the end of the Ed puzzle.